Good morning. Today the topic is sliding filament theory. Sliding filament theory, student, you can say this is explaining the complete muscle contraction, how the particular muscle will contract. Okay. So what actually happens? We all know that the initially signal will be coming from the CNS, or you can say the brain, and that signal will be brought to the muscle via motor neuron. Okay, motor neurons. It means the brain will send the signal. That signal will come in the form of motor neurons, and then it will reach to the muscles. And once the signal will be reached to the muscles, then the muscle will contract. Okay, and as we all know that what a single muscle is made up of many muscle bundles, or you can say fascicle. A single fascicle is made up of many muscle fibers. That is a kind of muscle fiber or muscle cell, and a single muscle cell is made up of many myofibrils. Now, what is myofibril? So, myofibril is nothing but a cylindrical structure. You can see cylindrical structure which has alternate actin and myosin bands, or you can say it is made up of actin and myosin filaments. Student, for muscle contraction, actin and myosin, these are certain structural proteins which are very important. Okay, and you can see in this case of muscle fiber, you can see in myofibril, the actin and myosin bands or the filaments are arranged parallelly and in alternate manner. It means this is actin, this is myosin, this is actin, this is myosin, like this. Okay. Now, for example, if this is the actin, and below this is the myosin filament. The upper one is actin, the lower one is myosin. What actually happens in muscle contraction? See, this actin filament will slide over the myosin filament. It means the only sliding of actin filament is observed, and that is why the name is sliding filament theory. Okay. Now, from exactly from which point of the brain or which part of the brain the signal is coming, that we will see. Okay. We all know that brain is divided into three parts: forebrain, midbrain, and hindbrain. Now, in this case, you can see majorly the forebrain is giving the signal for the muscle contraction. Okay, so here you can see this is the upper part of the forebrain. This upper part of the forebrain is known as telencephalon. Lower part of the forebrain is known as diencephalon. Now, it means this thing which you can see, this is the part of, you can say, telencephalon. To be specific, this is known as cerebral cortex. This is your cerebral cortex. Okay, cerebral cortex. So it is, you can see, the outer part of cerebrum. From there, the voluntary signal is coming for the muscle contraction, and you can see this you can say this structure which you can see, this is again both the structure are the part of the forebrain, but this thing is nothing but your diencephalon. Okay, this is your diencephalon. Okay, now. So basically, what I need to tell you is, if you see in the cerebral cortex, we have three regions: sensory region, association region, and then motor region. Okay, and these three regions are actually responsible to send a particular signal for the muscle contraction. Here you can see, here it is diencephalon. These two pillars which you can see, this is nothing but thalamus. Then at the roof you can see it is epithalamus. At the base you can see you have hypothalamus. Okay. Now this thalamus which you can see as a pillar or a gateway, actually it is just the gate to send the sensory and motor signal. It means if a sensory and motor signal has to be passed from the brain or away from the brain, then that thalamus will be acting as a relay center. Okay. So when the sensory signal is passed, when the sensory signal will go via thalamus, it will be first going to the sensory area. That sensory area will be passing the information to the what? To association area. This association area in cerebral cortex will process the information. Okay. Then after processing the information, it will send the signal to motor area. Now this motor area, student, is actually the final. You can say he will give the final signal for the voluntary muscle contraction. Okay. Now that final command or the final signal is coming from the motor area of the cerebral cortex, and it will come via thalamus, and then via, you can say, motor neurons, that particular impulse will be transmitted to the muscles. And once the muscle will take the info, you can say once the muscle will be receiving the impulse, then the muscle will contract. Okay. So here if you see properly, this is a single neuron, and here we have, this is what? This is muscle fiber, this is also muscle fiber. So these are separate muscle fibers which we have. If you see this point, okay, so this point is the point where your neuron is, you can say this is the junction where the neuron is meeting with the single muscle fiber. Okay, this is the point of attachment, you can say. So this is the junction, you can say, which is known as neuromuscular junction. This junction is actually known as what? Neuromuscular junction. Okay. So neuromuscular junction, you can say it is the point of attachment between a neuron and a single muscle fiber. But if you see this thing, now here a single neuron, you can say it is attached with what? Multiple muscle fibers. So this complete thing will be known as what? Motor unit. See, neuromuscular junction is the point of attachment between a neuron and a single muscle fiber. That is neuromuscular junction. It is also known as what? Motor end plate also you can say. And if multiple muscle fibers are connected to a particular neuron, that is known as, it is making a motor unit. Okay. Now, before going to the actual mechanism, what actually happens, you should understand some basic things. See, on any cell, you can say on any excitable cells, we have certain channels. We have certain ion channels. Okay. So we have certain ion channels. So ion channels can be of three types. You can say one is ligand gated channel, one can be mechanical gated channel, and one can be voltage gated channel. Now, very basic information, student. See, ligand gated channels will open when a ligand will bind. Okay, when a ligand will bind on the receptor, then this channel will open. Mechanical gated channel means what? These channels will open when there will be a kind of mechanical stress. And voltage gated channel means what? They will open at a specific voltage. Okay, if there is a specific voltage around the, you can say, plasma membrane, then these channels, voltage gated channel will open. Okay. Now we'll move to the actual mechanism. What actually happens? So here you can see, here we have a brief, you can say detailed explanation you can get here now. So before going deep inside the mechanism, you should also understand some basic structural things you need to understand. So you can see this is a motor neuron. For example, here we have series. From the series, the impulse is coming via motor neuron. This is a motor neuron first. Okay. And that particular impulse will be coming down. So here we have what? This is the muscle cell, or you can say muscle fiber. On muscle fiber, you can see these blue receptors are there. This is actually the acetylcholine receptors. See, this is the motor neuron. This is the end part. You can say this is the axon terminal, and here we have synaptic nerve. You can say this is the synaptic vesicle. Here we have synaptic nerve. This is your synaptic vesicle, in which you have neurotransmitter, which is filled. Now, this neurotransmitter is nothing but a common, you can take acetylcholine. Now, this blue thing is nothing but the receptor of acetylcholine here. Okay, here again we have one more, you can say kind of, this is ion channel for us, and then this particular plasma membrane of the muscle fiber is known as what? This is known as sarcolemma. Then this, if you see, the sarcolemma is actually enfolded. Yes, there is an enfolding of sarcolemma, which is known as what? This is known as T-tubule. Then you can see there is a sarcoplasmic reticulum. Student, sarcoplasmic reticulum is abundant, it is having abundant calcium. Okay, and calcium is very important for muscle contraction. This is the storehouse of your calcium, sarcoplasmic reticulum. And you see there are certain extensions of this sarcoplasmic reticulum, and these extensions are known as what? LTB. Okay, now you can see that there is a attachment between the T-tubule and L-tubule. This is very important, student. T-tubule and L-tubule is having a physical linkage, and that physical linkage is actually made by two proteins. This red one, which is your ranodine receptor, and this white one, which you can see, which is attached at the, you can see, which is from sarcoplasmic reticulum, it is attached. It is known as DHPR. Okay, dihydrogradient receptor. So this ranodine receptor and DHPR actually is making a physical connection between the L-tubule and T-tubule. Why it is important, that also I will tell you. Okay. Now here down you can see this is nothing but your myofibril. Okay, this is your myofibril. And this particular thing which you can see, this is the part of, you can say it is your subunit of troponin. Okay. So that's it for the structure. Now we will directly go with the mechanism. Okay. Now when this CNS will send the signal, that will be brought by the motor neurons. When the signal will be coming to this particular axon terminal, here we have certain channels. These channels will be the voltage gated channels of what? Of calcium. Okay. So when the signal will come down, so it at this point it will open the calcium channels. Okay, voltage gated calcium channels. Calcium will come inside. This calcium, student, will, you know, it will trigger the exocytosis of what? These synaptic vesicles. It means these synaptic vesicles will be fusing. They will be fusing here with the membrane, you can say with the membrane, and then your acetylcholine, this acetylcholine which you have read, right, will be released also. The acetylcholine which we have, this acetylcholine, student, will be released outside. And once this acetylcholine will be released outside, this acetylcholine will bind to its specific receptor. Okay, it is known as nicotinic acetylcholine receptors, or you can say this is again, you can say it is nothing but your ligand gated channel for sodium. Now once the particular ligand, that is acetylcholine, will bind on this channel, then what will happen? This channel will open and then you will see that sodium will enter. Sodium will enter the, you can say, muscle cell or the muscle fiber. Okay, now here what we have, it means the attachment. When the acetylcholine will bind to its specific receptor present on the sarcolemma, the influx of sodium is happening. Okay, now before the influx, the membrane potential which we have inside the muscle fiber is nothing but minus 70 millivolt, which is a normal membrane potential. Now after influx of sodium, this minus 70 can be shifted towards the positive value, that is minus 60. Now once if the sodium will come, positive ions will come inside the sarcolemma, you can say inside the muscle cell, it means the inside charge will become positive, and this will lead to the depolarization of sarcolemma. Now at minus 60, we have one more channel, that is VGC of sodium. This is very important, student. Ligand gated channel will be bringing your membrane potential from minus 70 to minus 60. But actually minus 60 will open the voltage gated channels of sodium, and this voltage gated channel of sodium will be bringing, you can say, sodium in huge quantity. It means the huge influx of sodium, which actually leads to the depolarization of sarcolemma, is due to voltage gated channel of sodium, not the you can say the ligand gated channel of sodium. Okay, now once the sodium will come inside, it means you have inside positive charge and outside you have negative charge. This is nothing but a depolarization signal, or you can say the action potential. Okay, now this action potential will be going via the sarcolemma like this, and then it will be going to the T tubules, which means the signal is coming down, and then finally it is coming here, the physical linkage. Now I told you that why there is a need to attach, why there is a need of this physical linkage. Student, see, the major aim is to pass the impulse to the sarcoplasmic reticulum. The very important thing is that you have to pass the impulse, it has to reach to the sarcoplasmic reticulum, because we need calcium from them. So now this impulse will be going down to the T tubules, and then finally from this physical linkage it will enter the L tubule. Okay, so from the T tubule it will go to the L tubule, and once it will go to the L tubule, here you have again some calcium channels. This again we have some calcium channels which will be opening here, so it means it is again VGC, voltage gated channels of calcium, and these calcium, and once this particular impulse will be reaching to the L tubule, it will open the calcium channels, and from there the calcium will be coming outside. The calcium will come in this sarcoplasm. This is the sarcoplasm, that is the matrix of muscle fiber. Now once the calcium concentration in the sarcoplasm will increase, once you have the calcium now in the matrix, that calcium can easily go and bind to the troponin protein. Now what happens further, that you can understand, but the only thing is that when the impulse is coming, the impulse is brought to the sarcoplasmic reticulum. Once the impulse is now reaching to the sarcoplasmic reticulum, the calcium will come outside. Once the calcium will come outside and you have maximum concentration of calcium in the sarcoplasm, that calcium will go and it will bind to this subunit of troponin protein, okay, which is associated with the myofibril. Now once the calcium will bind to the troponin protein, what happens, that you can see here. Now if you see this white filament, this is nothing but your actin filaments, and here we have the myosin filament. Okay, the yellow thing which you can see, student, here is nothing but this is your tropomyosin. Okay, this is your tropomyosin. This is the regulatory protein for us. This red thing which you have seen, this is troponin. Okay, this is what troponin for us. Okay, so if you see the actin filaments which are here and the myosin filaments which are here, which is just below the actin filament, it is actually, you can say, the interaction between the actin and myosin actually is inhibited by this tropomyosin. That is why tropomyosin and troponin, these two are regulatory proteins. If you see here, if I zoom this part here, you can see properly, this is your actin filament and here we have the myosin. Means this yellow thing is your actin filament and this is your myosin filament, and between the actin and myosin filament there is something like, there is some protein which is actually inhibiting the interaction. This interaction, you can say this inhibiting thing which is done here is done by a TPI. Actually if you see this complete thing, this is known as a troponin protein. Okay, this is a regulatory protein which will regulate the muscle contraction. So this troponin has three subunits: TPC, TPT, TPI. TPC means what? This is the calcium binding site. TPT means what? It will bind to the tropomyosin. See this tropomyosin. And TPI means what? This is the inhibitory subunit. It will inhibit the interaction between actin and myosin. Okay. But when the calcium will bind to the troponin subunit, that calcium binding will change the structure of troponin. It means some conformational change will happen for the troponin. It means when the structure will be altered, at that time this inhibiting unit which you can see, inhibitory unit which you can see will be flipped and the masking effect will be removed. See, in the presence of calcium, ATP and magnesium, if you get all three things, then this inhibiting sign will, you can say the inhibitory thing will be shifted and the masking effect will be removed. Now the actin and myosin is free to make a bond. You can say they are free to make a bridge. Okay. See student, I told you that actin will slide on the myosin. But for the sliding, first you have to connect the actin and myosin. For the connection, you require calcium, ATP, and magnesium. Magnesium is required for the as a cofactor, you can say. Okay. Now you understood for the calcium from where you are getting the calcium, and once the calcium, ATP, and magnesium will be made, will be required. You can say you will get. Then you can make a cross bridge. When the cross bridge will be made, once there is a connection between actin and myosin, so now the actin can easily slide, and that sliding thing you can see here. See. This is, you can say, the first thing. So if you start from here, see, when the ATP will bind, see, ATP will bind with the myosin. At that case, the actin and myosin is not connected. When the ATP will bind, this cross bridge, the cross bridge which I am telling you, this is the cross bridge now. Okay, so this cross bridge will not, it is not made. It means the cross bridge will break. This is very important. Cross bridge will break if the ATP will bind to the myosin, okay? And then once the ATP is binding, this particular myosin, actually this globular head which you can see of myosin has ATPase activity. So it can, you can say hydrolyze the ATP and it will store the energy of ATP. Now once the ATP is binding, this ATP they will hydrolyze and they will store the energy in this myosin head. Now this energy will be again utilized to make the cross bridge. And once the cross bridge is made, then the sliding will happen. It means here what happens? Here the cross bridge formation you can see. Here you can see the sliding. See, once you get the ATP, once the myosin will bind with the ATP will bind with the myosin, that ATP will be hydrolyzed. If you break the ATP, that energy will be released and that will be stored in the head of myosin. The same energy I am using to make the cross bridge. And once the cross bridge will be made, once there is an interaction between the myosin and actin, then after that interaction, this actin can slide on the myosin. And once this sliding will happen, this ATP and PI will be removed. Again, the site will be free. A new ATP will be bind, and once the new ATP will bind, again the cross bridge will break. It means cross bridge will break when a new ATP will bind. And once the new ATP is binding, then the myosin head will break the ATP and it will utilize the same energy to make again the cross bridge. Once the cross bridge is made, then actin will slide on the myosin. When the sliding will finish, again a new ATP will come, again the cross bridge will break. Clear? So this is something which is your sliding filament theory. I hope all of you understood this thing. Thank you.